What is phenotype? Last class we have discussed about in detail what is phenotype by Johansson experiment. Actually, when we start breeding, we have to start with some question. Like when you want to improve some trait, everything is start with some question. Then we have to breed for that particular trait. For example, you take any phenotype. You take a grain length or a flower color. You take flower color. You take flower color red. Red is dominant and yellow is recessive. The question we had to ask when you're choosing any, like for example, you want to improve any trait, you have to ask some questions. For example, you choose any one phenotype, flower color. Then first you have to think what is the genetics of that phenotype. Can anyone tell what is the genetics means? What question it is implies? Like what is exactly it means?
How many genes it is governed
Okay.
by?
Yeah.
By how many genes it is governed by?
Yeah. For example, this flower color having two alternative forms, red and yellow. Means you have to see which one is dominant, already written, but we have to choose which one is dominant, which allele is dominant, which form of the particular trait is dominant, and which form of particular trait is recessive. First thing, then in F1 only we will get to know this. Then in F2 we can see by based on the segregation pattern. I already discussed how to do with this. Based on the segregation pattern, we are going to know about either the particular phenotype is monogenic inheritance or polygenic inheritance. Oligogenic also may be possible, monogenic or oligogenic or polygenic. We have discussed in detail what is monogenic, what is polygenic, what are the characters of them. We will discuss in detail while I am dealing with quantity genetics. But this question you should know about when you are dealing with any trait, any phenotype. What is the genetics of particular phenotype? One is we have to see which form is dominant and which form is recessive. Then in F2 we can see either the trait is monogenic or polygenic. Then the next question is how many genes are governing for the particular trait? Okay, is monogenic or polygenic? Then how many genes? For example, monogenic only, digenic, trigenic. In polygenic also, in polygenic it's very hard to determine the number of genes is governed, like a particular trait is governed by, but we can have some uh, a rough estimate of like how many classes may be possible. Okay, we can't exactly determine how many number of genes if it is so governed by so many number of genes, but at least we can have a rough estimate. Then, after knowing that where the particular gene is located, we know that either the trait is dominant or not. How many genes are the like how many genes are controlling the particular phenotype? Then the question comes, where the gene is located? How we can detect? If I want to know
Map.
exactly, we also discussed this linkage mapping, cytogenetic mapping, physical map, how to create the linkage map. Okay, this is from that information we're going to get where the particular gene is, where the particular gene is there in the genome, gene on the genome by any maps. Can anyone tell which type of maps are can possible?
Cytogenetic map, physical map, linkage map.
Exactly. So we already discussed. I'm not going into it like with things physical map. We discussed in detail about linkage map. We will going to discuss while respective classes, cytogenetics and sequencing classes. Then, okay, now you know that dominant or recessive, how many number of genes, and you also know where it is located. For example, you find a gene in a one individual. What breeder one, you have to transfer into the famous varieties. Okay, for example, you identified one male sterility gene. Male sterility gene in wild abortive genotype, but you want to transfer into IR64 because we don't need all the characters of these genotype, wild genotype. Some varieties like varieties type of sulfan. For example, varieties glabri, varieties nivara, varieties whatever you take, wild genotype. Uh, but we don't want all the characters of genotype. We need that particular male cell trait in IR60. Then how we can able to transfer? Can anyone tell how we can transfer particular genotype?
back cross.
Only brackets? No, I want to improve the genotype, which having the both the character from the both the genotypes.
Pedigree also, that both if we
Any breeding method which involves hybridization. Okay, but you want to transfer the setup you want to transfer the only particular gene, but you want exact copy of IR64 with improved with having the male sterility trait, then you can specifically say the backcross method. Otherwise, any breeding method which involves hybridization, we can use to transfer the trait between the genotypes. Then, what are the factors you have to look into while transferring the genotype? What factors? We already discussed, you have to tell. What are the things we need to take care of while transferring the particular traits? We discussed in detail while discussing the breeding methods. When trait is monogenic, then which breeding method we have to what are the precautions we need to take? In which case, the transferring the gene from one individual to other individual or one genotype to other genotype is easier. If the trait is governed by
monogenic.
monogenic or a trait is governed by some few more major genes. If trait is governed by many minor genes, it's very difficult to transfer. You have to look if the trait is governed by a monogenic, which breeding method is feasible and which steps we need to additional steps we need to take care. If trait is governed by a polygenic inheritance, then how I can plan my hybridization and selection experiment. These things we need to take care while transferring any particular trait from one genotype to other genotype. Then the next question comes. Okay, you transferred, you created a hybrid. Okay, I'm not writing. This is the problem. Like admitting at late commerce, it's very difficult. Welcome, Nivedita. It's like our classes. Yeah. Then question comes. Okay, you transferred, you made a hybridization. The progeny will come. For example, you crossed P1 and P2. Then progeny will come. Think it as an F2. F1 is pass, self. In F2, you have to do selection. Okay, you know, either trait is dominant or not. I'm talking about everything is decided. Then you started your breeding method, like selecting the particular trait. Selecting the individual again are difficult. Selecting the desirable individual is the very important task. How selection of a trait will be decided? Like what are the obstacles while selecting what are the things we need to take care while selection for the particular phenotype? Can anyone tell? We also discussed while discussing about selection components. What are the things we need to take care while selecting the particular phenotype? Why are silent? We have discussed na, in detail. The class went for 2 hours.
Highly heritable, additive.
Yeah, we need to look whether the trait is heritable or not. How much variation it came in the particular population. When you cross the two individuals, how much variation it is created. For example, you are crossing male sterility is again difficult task. For example, tall plant into small dwarf plant. The plant is not only differ with the height, it also differ with the many character. Those who are unwanted from this plant and those who are unwanted from this plant, but population is having a recombination for all the traits. Okay, but we need to select the desirable plant height. But we want like 50-meter height, 100, we crossing 25, but we want 50-meter height as optimal phenotype. We have to search. But all the plants having recombined for all the phenotypes, not only the plant height. That's why selection for the particular trait is important. Then again, you have to know what is the behavior of the particular trait, the nature of the particular trait, how much heritability what the particular trait is having, how much easiness for doing selection. In case it is called a selection accuracy, how accurately we can able to phenotype the particular trait. Then another thing, how much variation is created, how much variation is there for the particular trait in the population. These are all the things we need to look when we have to do selection. If the trait is highly heritable, it's okay, well and fine. Can easily select. Easy for accurately we can able to phenotype. For example, flower color. Uh, it is visible to our eyes. Okay, we can easily select the flower color. But if you want some resistance, blast resistance in rice, we had to inoculate, we had to screen it, we had to score it. It's very difficult. Then, if it is no variation present, how to do selection? This is all the things we need to take care while choosing selection while you are doing selection for a particular trait. Then, how many years it will take to improve the particular phenotype? This is also one of the important question we need to ask ourselves as a breeder before going to improve any traits. We already discussed how trait like time is also very important. For example, if it is a quantitative trait, it will take very long years to improve that particular phenotype. If it is governed by a very few major genes, it's very easy to develop a particular variety with a small number of years. Like it is a very less number of years. These are the questions we already discussed. That's why I'm not going in detail. In 7 to 8 classes, like 5 to 6 classes, I discussed these things only. But how to correlate them? This is the summary. Why I taken this much lengthy hour of classes for each and every point? Because those are very important while doing a breeding for any particular phenotype. Now you have, I think now you got a clear picture about why I have discussed those things initially, rather than discussing many things in detail. First, we have to look at the genetics of the particular trait. How many numbers it's governing, like how many number of genes are governing for the particular trait, where the particular gene is located, how much easier it is transfer the particular gene from one genotype to other genotype, what are the hurdles, pros and cons of while doing a selection for the particular phenotype, what are the things we need to look, then what is the time I required for the improving of the particular trait. These are the main thing we need to understand before going to the breeding of any phenotype. Okay, now you are clear with this.
Yes, sir.
Then the other thing, No need of marker if we have this. For example, I jumped. Actually, one or two slides I deleted unconsciously. Sorry for that. Yeah, we'll discuss this thing. For example, you want to improve particular trait. It is take yield. Yield having very low heritability, and yield is a dependent trait like which are dependent on so much independent traits. I hope you are getting what I am saying. What is independent? What is dependent trait? Independent trait like plant height, grain number, panicle length, the number of grains per panicle, all the traits which are in chlorophyll content, the leaf shape, leaf area, everything is an independent trait where our yield is dependent on. Okay, like where yield is dependent trait, which depends on these independent traits. We had we had to improve these traits, then indirectly we are improving the yield. This is what all your everyone is doing in MSc, like checking the path analysis, correlation analysis, all the things we are doing for this only. See, if you want to improve the yield, you have to check all the traits which are related to yield. If you want to improve means how many traits you have to look at a single time. Breeder don't have 1,000 eyes. They have only 2 eyes and he having 2 hands only. How many plants he has to observe and how many traits he can observe at a single time. It's possible to observe all the traits at a time in such a large number of population. How much time it requires to improve the particular trait if it is governed by so many number of component traits, or if it is governed by so many number of genes, the quantitative inheritance. Why the breeding for quantitative inheritance is very difficult? Can anyone tell in 2 sentences? And it is there in the slide. I will skip this. Why? See, everyone tell breeding for quantitative trait is very difficult. Why? Why it is so? Can anyone tell two-line answer for that?
Because many genes go on it, sir, and it
Okay,
is
many
affected by
genes is governing.
environment.
What happens? If it is governed by genes, see in major gene also, you are not going to look at the genes. We are going to look at the phenotype. But if you
It
are
is
The
affected by environment, sir.
okay, one
heritability
thing.
is low also.
Why? See, male sterility is governed by how many number of genes normally? Like how many number of genes?
One,
Male sterility.
one,
One.
sir.
Can you heard about environmental sensitive genetic male sterility, photosensitive genetic male
Yes, sir.
sterility?
Yes, sir.
It
Yes.
is also governed by one, but how much environment is affecting particular phenotype? If we put the same genotype in high temperature, it will become male fertile. The same genotype if you put in like a low temperature, it will become sterile. Like take it inverse. Sorry, I'm mistaken. See, it is the trait is even though it is governed by a single gene, it is affected by the environment. If you change small temperature, it will change the phenotype. Fertile to sterile, sterile to fertile based on the dependent temperature, even though it is governed by single gene. How environment will affect the trait? We will discuss while discussing quantitative trait. But as of now, why breeding for a quantitative trait is very difficult? Tell me in 2, 3 answers. Yeah, if environment is good, then other than the environment,
The heritability is low.
yeah, exactly. Heritability is low. Then another thing, generally you say in field you are going, breeding for that particular trait is governed by many number of genes is very difficult. Quantitative traits are very
We
difficult to
can't
deal.
classify them as clear-cut classes, sir. Clear-cut
This is
differences we
what
don't know.
this answer I am expecting actually. See, if it is governed by a single gene, you will get a clear cut classes. In field, you will get, for example, this row having like in field you can classify if 100 plants is there, you can easily classify 75 into 25. If it is governed by a single dominant gene or a single recessive gene, we can classify. Okay, 75 this particular plant, 25 is recessive plant. Like that, we will classify. Okay. But in case of trait is quantitative in nature, you will get so many number of classes. And as I said, it's affected by environment also. Environment effect is another thing. Like I will explain why environment effect is more in case of quantity trait. This is the one question I will going to explain while dealing with quantity traits. See, how many number of classes are? You have to screen all. Selecting from this large variation, selecting the desirable trait is very difficult task. You have to screen all the population. You have to check the measurements of all this one. See how many classes for one gene you are getting so many class with two class. Okay, 3 power n like genotypes. If you are like if it is going to be 25 genes, 3 power 25. How many phenotypes? How many genotypes you are going to get? 4 power n. If number of genes increases, how many phenotypes you are going to get? And selecting your desirable phenotype from the particular population, that become a very difficult task if it is trait is governed by many number of genes. That's why breeding for a quantitative trait in the breeding for the phenotype which governed by a quantitative trait inheritance, it's very difficult. One is environmental, one is we can't make a clear classes. And there is a disadvantage in this also. In clear this while selecting this monogenic trait also sometimes it's become difficult. I will explain in detail while discussing about gene for gene hypothesis. Yeah. See why I'm discussing all those things means, for example, improving yield is a difficult task because you have to improve all this. But there are if you want to improve yield, this yield may be linked with some of the traits which are easily phenotyped. For example, you take, I will discuss in detail. They are called as proxy trait. I will discuss this slide later. Okay. See, if there are two individuals. You can classify them easily because this flower color is white and this is color having a pink like purple. We can able to classify them very easily. We can selection for the flower color is very easy by looking. See, by looking from the slide only we can able to classify them. For example, you take resistance. These two individuals, like many individuals, like by looking at them, you can say which plant is resistant, which plant is susceptible. It's possible.
No, sir.
No, it's very highly. You have to give inoculation, then you have to observe, you have to keep them in a particular environment so that your disease can develop. Then after then you have to screen them, score them. Then you have to say that okay, this particular plant is resistant and this particular plant is susceptible. But if it is a flower color, if it is a flower color, we can easily classify. Why it is so? Because it is visible to our eyes. Okay, the trait is visible for our eyes. That's why it's easy to classify them into two different individuals. But the resistance is it's invisible and it needs some congenial environment. It has to develop some symptoms. Then only we can, even the symptoms are, if it is gone by many genes, then again we are having a continuous stress. One is 5, 4, 3, 3. I think many of you done like scoring of the particular disease. See, if the trait is very complex, it's very difficult to select. Then how can I select the resistance? Easily. What are the possibilities I can make as a breeder so that my life will become easy to improve the resistance? What are the things I can think? See, many of you are, I think majority in my classes already done MSc. Like 70% are MSc holders. You should tell the answer. That's why I'm asking questions.
Linking of phenotypic data with genotypic data.
Think that I didn't told about any genotypes. See, genotypic data, how much I had to run the gel, I had to see. Okay, genotypic data is one thing. Tell the basic, come from step by step. I don't have any genotypic data. I don't have sequence of this. See this. I don't know even flower which crop I have taken. I don't know the reference genome, or I don't have any genome sequence of this particular species.
Presence of any particular phenotype in the plant having resistance and absence in susceptible plant.
Can you refine your what you want to say? Like, can you refine it in a proper way?
Presence of any particular trait or any particular morphology which is present in resistant one and absent in susceptible one.
Yeah, yeah. Okay, you are missing one term. Your understanding is correct. You are understood rightly. Yeah. What he said.
Sir,
Yeah, tell.
sir, we can make classes like highly susceptible or less
No,
susceptible.
I told that is difficult. I had to inoculate, I had to screen, I had to score, then I had to check. It takes a lot of time. But I don't want to do all those things, but I want to select directly which one is resistant, which one is susceptible in the field. Any possibilities. See, every possibility will not work out at every situation. Just think any possibility. You are already a masters in breeding. Many 90% of my strength. You are all breeders. You are all competitors for me also. Like,
Sir, if the plant survive, then we can classify it as a resistant plant.
plant, for example, it is a obligate trait. If plant is not survived, this is also will not going to survive. Then what can I do?
Sir, I didn't understand. What did you say?
For example, if the disease is obligate in nature, if plant is disease will not occur, like disease will not going to survive if the plant is not there. If plant is not in living condition, like virus to take.
Okay, for its survival, it makes the plant to live. That is what you're saying.
Yeah. It's an obligate in nature, like obligate in nature. The
Sir, we
survival is
can measure the yield, sir. Which variety is giving the
the
yield?
yield is another difficult trait. Say saying yield is affected by how many number of independent traits. For example, these two plants having one plant is having less grain filling and another plant is having low grain filling. That also affect the yield. Then how can you say that the resistance only making the better yield? Think simply, like first you start with something which is easier. You have to make your life easy, not difficult.
The plant which is showing more symptoms.
See, symptoms, okay, if this is not showing any symptoms, it having hidden symptoms. Even some, it directly makes the death of the plant. Think actually, see, I told already, for example, you take this flower color, white color flower linked with your resistance. Then means the plant having, which plant is
It will
having a
go for
white color?
indirect selection.
Exactly, indirect selection. Okay, that's what I am asking from you. Okay, why I'm asking so many questions? Because all you are all MSCs. You should have able to answer. Okay, because if you don't, it's when the situation arises, the direct selection for the particular trait is difficult. All breeder will look into indirect selection. Okay, any proxy trait can I look? So improving that trait can selecting the trait maybe indirectly selects other trait. For example, if you are taking a white color flower, there are so many examples. Taking a flower color, they may be white color are resistance, purple color are susceptible. Means that if you select white color flower plant, you are also selecting a resistant plant. If you are rejecting purple color flower plant, then you are rejecting all susceptible individuals. It is called as an indirect selection. Means selection for a flower color indirectly selects for your resistance. This is called as marker assisted selection. In this case, your marker is
Flower color.
exactly. Your marker is your flower color. Means your flower color aids in the selection of your resistance. So you need not to go any inoculation, no need to develop any disease symptoms, direct selection. See how much your life will become easy. If you identify any trait which are easily screenable, which can detect easily and we can classify the individuals easily, and it also selects for your desirable trait. Means this is called as marker aided selection. That's why we need marker to make our life easy to select complex trait. That's why we need a marker in the plant breeding. That's why nowadays everyone is fall behind the marker assisted selection. It started with in 1923 itself, I think. Sachs experiment, where the seed size is related with the seed color. If you improve seed, like so for example, there are some seed color, for example, white seed color having larger seed size, blue color seeds color having smaller seed size. Like that, the color of the seed is linked with large, like size of the seed. Flower color may be linked with the resistance. One classical example in rust, I think, the burning or the burning symptom
Tip
at the
necrosis.
tip
Tip
is
necrosis.
yeah, tip necrosis is linked with your rust resistance. There are several examples.
LR24.
Yeah, there are several examples. You can select for the particular trait, which will indirectly select for your complex trait. Understood what is marker assisted selection, and understood why we need a marker in the plant breeding. Any doubts regarding this?
No, sir.
Any other? Because from day one, I'm receiving only 3-4 response and the same voice. Even I can recognize voice also. Any other than you? Deblina, you are there?
Yes, sir.
You understood what I'm saying?
Yes, sir.
Santoshini?
Yes, sir. Understood.
Mandeep?
Yes, sir.
Yeah. Manikarnika? Understood?
Yes,
Understood?
yes, sir. Yes.
Very Soumyajit, understood or not?
Yes, sir.
Okay. And ask the doubts. If you have any doubts, ask in the class. Okay. No need to hesitate. I think you are in MSc first year, no?
Yes, sir.
You have so much opportunity to learn. Okay. Don't hesitate to ask doubts. Not only for me, for your teachers also. Okay. Whoever come for the class, you just ask any doubts. Okay. You are still in learning. Okay. Then, okay, then I will come to this thing. That's why. No need of markers. I told about what is marker aided selection. We don't need marker if we have this If its trait is easily scored, I told what is the importance of that. If the individuals were reliably classified into few distinct phenotypic classes, and more particularly means it's a monogenic many times. There were a complete correspondence between the trait phenotypes and the genotypes of the conserved loci. It means that P is equal to G plus E. If environment is having more effect on the phenotype, you can't able to select easily. For example, take a disease one, blast. One of my batchmates is conducting the blast screening experiment in winter. Blast needs higher humidity. In winter, we hardly find low humidity is there. Like in summer, we'll find higher humidity, but she is screening the rice in the winter. She can get easy, like reliable screening is possible. No disease development happen. Okay. The entire experiment is collapsed. If she having any marker which linked with this particular disease, for example, you take flower color only. Based on flower color only, she can select the plant, even though there is no need of any inoculation. If it is environment plays a very important role in the manifestation of your phenotype, then you need a marker. But if you don't have any environmental effect, if P exactly is equal to Z genotype, that there is a complete correspondence between the phenotype and genotype, and there is no environmental effect, then we no need of marker. Okay, are you clear with this? What condition we don't need marker? What condition we need a marker? Understood?
Yes,
Anybody
sir.
have doubt in this? Okay. And don't later you don't ask that I understood. I have not understood. See, that's why I'm repeating. And those who are listening recorded classes also, try to attend live classes. See, okay, if some excuses are allowed, but okay, try to attend live class so that you have an opportunity to ask the doubts in the class itself. Okay, because I was doing something, some directly WhatsApp message will come, explain about some marker, explain about RFLP, explain about SSR. It's difficult to answer. We can't make a single line WhatsApp text to like convince you. It's very difficult. I need to type a essay for you in the WhatsApp and making understanding the difficult thing in the WhatsApp chat. It's very difficult. So that if you want to learn, make see I'm announcing the time is fixed 9:30. If you want to learn something, you should make the time. Then only you will learn. You're going to learn. Okay, it's not like that. I will attend recorded classes. I can text at any time. Many times I'm not going to reply you. Okay, like if some doubts are there, it's easily it can easily clarify in the WhatsApp text. I can answer. Some topics are which are very difficult to explain in the WhatsApp text. I can't answer. Okay, please if you want to learn, attend the live class. Then ask the doubts here itself. Okay. Then there is a concept of polymorphism. Before I am going to understand anything about marker selections, markers, types of markers, we should understand what is polymorphism. There is a word, it includes polymorphism means occurrence of multiple forms of a trait for a gene within the population. For example, this population having some variation, like take flower color. This is a polymorphism, white color flower. If it is a more color, it is again highly polymorphism. Means it's a common phenomenon in genetics. Means every gene having an alternative form. This is called as polymorphism. I think many of you are already done. So many are dealing with variability studies. You are already variability study means you are looking at the how much polymorphism in the particular population. Then what are the different types of polymorphism? It's existed in the population. One is naked eye polymorphism. This is called as naked eye polymorphism. With your naked eye, you can able to classify them into two different individuals. This is called as naked eye polymorphism. It means the morphological traits that were the earliest genetic markers, they are used in the scientific studies. For example, flower color, seed color, or anything. Fruit size, fruit color, fruit shape, like some are star shape, some are elongate shape. Any observable trait that can easily score with a naked eye, that is called as naked eye polymorphism. Then another type of polymorphism also possible at the biochemical level, at enzymatic level, called as biochemical polymorphism, such as isogymes, allogymes, okay, like protein markers, enzymatic markers, some some days they're using different biochemicals also a marker, some metabolite, presence of some metabolite or absence of metabolite. These are all the biochemical traits we are using for detecting the polymorphism. Then at last, ultimate is your molecular polymorphism. Means there is a variation exists at DNA level. Is called as the polymorphism at molecular level. Broadly, we will say all these enzymes and all these things molecular polymorphism, but exactly it's like naked eye polymorphism, biochemical polymorphism, and molecular polymorphism. We will discuss in detail one by one. Okay, directly jump. Okay, for naked eye polymorphism, it's simple, and there are so many traits. Like as it tip burning is resistance with tip burning associated with your rust LR34. As of now, I remember this only one example. Sorry for me. Like for that, there are so many examples which there in the AKSing and BDSing for the naked eye polymorphism. You read it. Then biochemical polymorphism. I will discuss in detail. Yeah. Before going to biochemical, I want to discuss the DNA polymorphism. Actually, some slides are missing. Like I don't know, some error is happened. Yeah. What is polymorphism, Viraj? The polymorphism at DNA level can be possible at so many types. Either the polymorphism at single nucleotide level, polymorphism based on there is an A insertion or any deletion of the particular base or a particular sequence, or there is a variation in the copy number. You will discuss in detail what is copy number, what is insertion and deletion, what is SNP. Just remember at DNA level, there is existence of polymorphism at single nucleotide level, either because of insertion or deletion of some base pairs or some sequences, either because of there is a variation in the copy number of the particular DNA segments. Like it is called as repetitive DNA. Mainly, this copy number variation observed in the where the DNA is repetitive so many times. Okay, there are microsatellites, many satellites we will going to discuss. Then, what is genetic marker? A trait that is polymorphic. This is a important. Okay, we're going to that. I have to. What is genetic marker? Anybody have? Please define. They have already given SRF. You should tell. What is DNA marker? Sorry, what is genic marker? I told all the things. You should just put in a proper way. Try at least.
Any any trait which is linked with uh, phenotype, not genetic trait, character. Any trait which is linked with a character.
Any trait is linked with the. This is what definition marker. I told a very beautiful story. Okay. If any trait is linked with a character, it is just defines, it justifies the definition of genetic marker. I told a very different, like beautiful story, how to select, what are these things. What is the lack in your definition? Can you at least guess? Anybody guess what is the lack? What is lacking in his definition? Any traits that is linked
Any
with the character.
easily identifiable traits, sir.
Okay, easily okay. When you define, some things must be compulsory. See, everyone reading the same books, but everyone getting the same rank. This is the difference. While writing the answer, you should make some difference so that while telling any answer, you should convey some meaning. You told any trait that is linked with a character. What can I do with this? If any trait is linked with any character, what can I do with this? I told the things which I told about
Any
polymorphism.
easily identifiable trait, by selecting it indirectly we can select the desirable characters.
Yeah, that is actually half definition. Okay, at least you are on the way. Okay? Until you not used for somebody left the meeting. Okay, let it. Somebody like means you are in the halfway. Okay, there is some missing. I will tell. See, any trait that is linked with the character means there are linkages always there. Okay, most of the traits. See, chromosome means it having only single gene, having so many genes. The genes present in a single chrome, they are linked only. Until unless it is not used as a marker, then how can I say this linked, any trait is linked with a character? How can you make this thing into a, how can you bring this thing into a definition of genetic marker?
Closely linked.
Closely linked also. Until and unless, if I not used as a marker, then how it will become a marker? Okay? That's why while defining, don't say like you should have some meaning. Okay? What is marker? We'll discuss. A trait that is polymorphic. For example, if the both plants having white color, can it be a marker? By using this flower color, can able to distinguish two individuals? It's possible or not?
Yes, sir.
Yeah. How it is possible? The,
Different color, different genotype.
I told both the genotypes having a single white color. Then the flower color can be
Then
used
not
as a
possible.
marker or not? What?
Yes.
No. Means the trait is monomorphic means you can't be used it as a marker. Means you should first satisfy the criteria of polymorphism. Polymorphic. That's why I explained the slide. Polymorphism means if a trait, if you are using any trait as a marker, you should follow some criteria. One is first thing is polymorphic. Those who are already done any marker studies, I think many of you know, first thing we are doing is polymorphic survey. Okay. The trait is must be polymorphic. This first criteria we need to follow. Then easily and reliably identify. This is the another thing. If the particular trait is itself is a difficult to phenotype, then what is the need of using it as a marker? Marker is used because to select easily for the complex trait, like indirectly selecting the complex trait. But if a trait is difficult to phenotype itself, then what is the use of using this as marker? This trait is the marker. No use. That's why first criteria should follow polymorphic. It should be easily and reliably identified, and it is readily followed in segregating generation. Can anyone say what is the meaning of this line? This line I directly took from MA's book by Ekesing and BDC. Is readily followed in segregating generation. And should segregate.
Sir, it can be identified in segregating generation, easily identified in segregating generation.
That point I already repeated, already wrote. If the both the convey same meaning, then okay, why should I write?
One generation to the next generation to the again next generations.
No, you should pass means you should have high heritability. There will be one question in SRF only. The marker should have 100% heritability. For example, your molecular markers like SSR, all those things having 100% heritability. Okay. And to segregate. For example, giving different bands, then only you can be able to select. Okay. Should follow, should having 100% heritability, and it should segregate in the generations. If it is not able to segregate, any segregation distortion is there, then you're not going to get anything. That's the meaning of this. Means you just remember, trait is polymorphic, easily and reliably identified, and is readily followed in segregating generation. Indicates that, and it should also exhibit, if all the trait is confirmed, then it is known as a genetic marker. Then, what is the properties of an ideal marker? Means if you are using some trait as a marker, then what are the ideal characters should must have? Means I told polymorphic. I think many of you are okay with this. What is polymorphism? Why we need polymorphism to use it as a marker? I think everyone clear with that. Then you should having multi-allelic image. For example, you have only two flower color. Okay, you can classify them as two. For example, resistance, moderately resistance, highly resistance, low resistance, like this, so many classes are. Your resistance is linked with the white, for moderately resistant linked with purple, highly resistant is linked with some green color flower. Means how many polymorphism for the particular trait is there, it is you'll getting the chances of using that as a marker, it's very high. Means ideally marker should have more alleles. I will explain while explaining SSR or any other molecular marker. Why particular marker should have more alleles so that you can select better. Should now, as of now, you remember should be polymorphic and it should be multi-allele. Means you should have more number of alternative forms. If you are having more number of alternative forms, you are having more polymorphism in the particular population. It is easy to select. Then it should be codominant. I will come while explaining dominant marker, codominant marker, what are the traits. Okay. As of now, you remember marker should be codominant, polymorphic, multi-allelic, and should not be epistatic. I explained in detail about epistaticus, you should answer. Why marker should not have any epistaticus? Why marker should not be epistatic?
Sir, if it is epistatic, then it cannot be easily expressed in next generations.
See, I'm not talking about expressivity. Not penetrance, expressivity, I'm not talking. I'm talking only about epistatic. What is that? You should know what is epistatic. I think I discussed in detail what is epistaticus.
Sir, it will interact with other genes, so we cannot identify it easily.
Exactly. This answer you should tell. Means, for the particular trait is epistatic with another trait. Then how can you identify? How can you use it as a marker? I told marker should be linked with your desirable trait. But the marker is also epistatic with other another trait which are not useful for you, simply confuses your selection process. That's why your marker should not be epistatic. I am not going in detail because I already explained what is epistatic. Then the marker loci should be neutral so that alleles by themselves do not affect the fitness of an individual. What exactly this statement means? For example, if our flower color is affecting the genes which are affecting flower color may be associated with the fitness of a particular individual. It means that white color flower individuals will not going to survive. Only purple color individuals will going to survive. Means the particular trait is having some role in the fitness of an individual. For take grain size. If you're using grain size as a marker, grain size will definitely affect the fitness of an individual. If you're using that as a marker, you will selection procedure will also biased. That's why if you are using any marker, it should not directly correlated with the your survivability or fitness of when a genotype. Understood what this point means?
Sir,
Marker should be neutral. It should not affect with the fitness or survivability of your individuals. Okay, understood? Any doubts?
sir, you said that grain size can affect the fitness of the individual. I didn't understood that, sir.
Yeah. For example, if marker is one trait you are selecting as a marker. But that marker is very much essential for survivability or fitness of an individual. Means you can't use this trait as a marker because it will affect. If it, for example, if you're one color flower, white color, if the flower, white color flower, having some sterility issues, but purple color will fertile in nature. I can use it for sometimes, but as of now, means the fitness of the individual is affected by this particular trait. Means you should not use it for the marker. For example, you take molecular marker. This is actually one of the disadvantage of the naked eye polymorphism. That's why we are going for a molecular markers. For example, you take SSR marker. This marker genotype is nowhere related with your fitness of an individual because they are repetitive genes. There are so many they are mainly in repetitive genomes. They are not having any exons. They will not affect any particular genes. That's why you can use it as easily as a marker. But any trait which affecting the survivability or fitness of an individual, you can't use it. Understood?
Yes, sir. I understood that, but grain can affect the fitness of a individual.
Oh, yeah. So sometimes, okay. For example, if having a larger grain size, so the amenable for the damage is very more. Maybe viability is also one of the factors. If larger seed will not viable for many times, means it will not going to survive for next generation. Yeah, it is not maybe affect the fitness of the parental individual. Maybe progenies will going to affect. Okay. But just for example, I said, if any trait is affecting the fitness of an individual, you can't use it as a marker. Many cases, grain size will not be related with the fitness. That also happens. I'm not telling grain size is involving in the fitness. Okay, it's not like that. For example, like that. Okay,
Okay,
pointed
sir.
rightly.
Okay,
Yeah,
understood.
but I'm just taking an example So that if any trait which are involved in the fitness of an individual, you can't use it as a marker. Okay. Now you got.
Yes, sir. Yes, sir.
Not like that brain cell is always involved in the fitness. Like, yeah. This should be abundant, distributed almost evenly over entire genome. I will tell this point when I will discussing with the DNA marker specifically. Okay? You should have abundant. You should have abundant polymer. Where with this limitation of a biochemical trait, like where this morphological trait. See here only two classes are possible. In flower color, in this case only two classes are possible. But there are these two classes only can't able to decide your resistance and susceptibility. Then we should go with another option so that we can able to identify resistance and susceptible plant. That thing will comes under your molecular markers so that it having so much advantages over the all other markers. I will explain which are the types of marker systems are available. As of now, we have available with these types of marker. One is morphological marker, another one is protein marker, another one is DNA marker. Morphological markers are naked eye polymorphism, we discussed. Protein markers I will discuss. DNA markers I will discuss in detail. Okay. We attend live classes for these markers because markers are very tricky. Until unless you will show an interest, you will not going to understand marker class. Okay, any doubts up to this?
No, sir.
We
Yes,
are ending the class here. Okay. So that
sir. You are
means
going to
you
continue.
understood what is the genetics of polymorphism, what is polymorphism concept, and types of polymorphism. Okay.
Yes, sir. Yes, sir.
Then any doubts?
Sir, you will continue markers, sir. Means you will first take markers and after that you will take the plant breeding. Means you said that you will first complete the plant breeding methods and everything.
Yeah, actually, no. We'll start with the markers. Along with the markers, only see. Actually, plant breeding is associated with the molecular breeding. For example, take marker assisted backrest breeding. If I want to teach marker assisted backrusting, first I need to teach backrest. Okay. If I teach all the breeding methods now, again, if I want to explain marker assisted backrest breeding, I need to repeat the things again and again. That's why I will start with the markers. I will cover what are the different types of markers, what is mapping, what is QTL mapping, all those things. Then I will start with marker assisted breeding. If I start with marker assisted breeding, you should know what is the breeding methods. By in and like while covering these things, it will also cover the breeding methods also. That's why I planned the lectures like that. Okay.
Okay. Okay.
But that's why I will cover up to Jivas. Then I will start marker assisted selections. There I had to use recurrent selections, background selections, pedigree selections, single cell design. Okay, by using markers, I will explain conventional and advanced, and I will compare the both at the time. Then it is easy to learn.
Okay.
Okay. I will club the classes. Okay. But as of now, I will finish up to Jivas. I will take continuously. Then after I will come to quantity. Okay. Any doubts up to this? Either I will learn. Right?
No, sir.
Good night.